Hello everyone, I'm Dr. Malik M.M.Siddiqui. Welcome to my YouTube channel. Today I'm going to start a lecture series on immunology, that is understanding immunology. And in this series, my lecture 1 is concerned with immunity. First of all, let us have some introduction about the immune system. The immune system is a versatile defense system that has evolved to protect animals from invading pathogenic microorganisms. As we know that on daily basis our body is encountered by a variety of microorganisms like bacteria or virus, etc. Some of them might not cause any harm to the body, but the others can be pathogenic, that is, they can be disease causing. So, it is the responsibility of our immune system to generate enormous variety of cells and molecules which will be capable of specifically recognizing and then finally eliminating those foreign invaders. Now, the word immunity comes from the Latin word immunis, which means to exempt. So, that means the English word immunity means the state of protection from infectious disease. Now, functionally, an immune response can be divided in two related activities. The first activity is the recognition, and the second one is the response. The immune system is able to recognize subtle chemical differences that distinguish one foreign pathogen from another. So, our body, our immune system is capable of recognizing even many very minute differences in one pathogen that are not present in another pathogen. Now, once a foreign organism has been recognized, the immune system it recruits a variety of cells and molecules to mount an appropriate response. This response is called as effector response, and this response is to eliminate or neutralize the organism. So, in this way, our immune system is capable to convert the initial recognition event into a variety of effector responses, and each of these effector response is suited for eliminating a particular type of pathogen. That is to say that the effector response can vary upon the particular type of pathogen. So now we know that immunity is a state of protection from infectious disease, and this immunity has got a less specific and a more specific component. Or in other words, we can say that immunity can be classified in two different types. Number one, which is called as innate immunity, it is less specific, or we can say it is a generalized kind of immunity. And the second one is called as adaptive or acquired immunity, but it is more specific. So the less specific component, or which is innate immunity, it provides the first line of defense against infection. So it is a generalized response against the infection. It is less specific. Now the phagocytic cells such as macrophages and neutrophils, and the barriers such as skin and a variety of antimicrobial compounds which are synthesized by the host cell, they all play an important role in the innate immunity. The second type of immunity, which is called as adaptive or acquired immunity. So adaptive immunity responds to the challenge with a high degree of specificity as well as the remarkable property of memory. Typically, there is an adaptive immune response against the antigen within 5 or 6 days after the initial exposure to that antigen. The major agents of adaptive immunity are lymphocytes and the antibodies and other molecules produced by them. Now, some details about the innate immunity. Innate immunity can be seen to comprise of 4 types of different barriers. Number 1, anatomic. Number 2, physiological. third phagocytic, and number fourth is the inflammatory. Let us have some knowledge about anatomic barriers. The first in this series is the skin. As you know, skin is the first line of defense. It is a mechanical barrier which retards the entry of microbes into our body. Secondly, the acidic environment which is present, that is between pH 3 to 5, it is also retarding the growth of microbes. The second barrier, anatomic barrier, is the mucous membrane. In mucous membrane, you have got a normal flora. So the normal flora will compete with the microbes for the attachment sites as well as for the nutrients. Now we have got mucus. Mucus is the entrapment. So the foreign microorganisms, they can be entrapped in the mucus. And we have got cilia. The cilia are present to propel the microorganisms out of the body. Second type of barriers are physiological barriers. For example, number one is temperature. Our normal body temperature, it can inhibit the growth of some of the pathogens. And the fever response, it inhibits the growth of some of the pathogens. Then low pH can be cause of retardation of growth of the pathogens. And this can be seen in case of the stomach. For example, we have ingested orally some microorganism, but when it reaches to the stomach, the acidity of the stomach, it will kill most ingested microorganisms. Now third one is the chemical mediators. So various activities can be done by some chemicals or enzymes. For example, number one is lysozyme. Lysozyme is the enzyme which cleaves the bacterial cell wall. So the bacterial growth is retarded. The bacteria are killed. Now interferon, the interferon induces antiviral state in uninfected cells. Then we have got complement system. The complement lyses the microorganisms or facilitates the phagocytosis. Then we have got cytokines. These are toll-like receptors which recognize the microbial molecules and then signal cell to secrete the immunostimulatory compounds which are called as cytokines. Then the collectins are also performing the function of disrupting the cell wall of the pathogens. Now the phagocytic or endocytic barriers. There are various cells which can internalize or endocytose and break down the foreign macromolecules. The specialized cells like monocytes, neutrophils, tissue macrophages, they can internalize, that is, they can phagocytose, they can kill and digest whole microorganisms. Next barrier is the inflammatory barrier. Now the tissue damage and infection induce leakage of vascular fluid containing serum proteins with antibacterial activity and influx of phagocytic cells into the infected area. Now comes the second type of immunity, that is acquired or adaptive immunity. Adaptive immunity is capable of recognizing and selectively eliminating specific foreign microorganisms or molecules. These molecules, they will be called as foreign antigens. Now the adaptive immunity displays four characteristic attributes, four important features. Number one is the antigenic specificity, number two, diversity, number three, immunologic memory, and number four, self and non-self recognition. Now the antigenic specificity of the immune system permits it to distinguish very minute differences among the antigens. The antibodies are capable of distinguishing between two foreign molecules or two protein molecules that differ only in a single amino acid. So you can see this minute difference can also be distinguished by antibodies. Now the second attribute or feature is the diversity. The immune system is capable of generating tremendous diversity in its recognition molecules. So this will allow it to recognize billions of unique structures on the foreign antigens. Once the immune system has recognized and responded to an antigen, it exhibits immunologic memory. This is a very important feature of the adaptive immunity. That is, a second encounter with the same antigen induces a heightened state of immune reactivity. Because of this attribute, the immune system can confer lifelong immunity to many infectious agents after an initial encounter. Finally, the immune system normally responds only to foreign antigens, indicating that it is capable of self and non-self recognition. As we know that inside our body various protein molecules, various enzymes they are produced, but our immune system is not producing any antibodies against those proteins or enzymes which are present in our body. So they are recognized as self molecules. And the other foreign particles, other foreign antigens, they are recognized as non-self. So our body is producing antibodies for non-self antigens. In case of autoimmune disorders, you will see later on that our body is producing the antibodies against some enzymes or proteins which are the part of our body, which are self molecules. So the ability of the immune system to distinguish self from non-self and respond only to non-self molecule is very essential. Now, this kind of adaptive immunity is provided by humoral and cell-mediated immunity. That is, the adaptive immunity can be achieved by humoral or cell-mediated immunity. During the 1930s, mainly through the efforts of Alvin Cabot, a fraction of serum, which was first called as gamma globin and it is now called immunoglobulin, was shown to be responsible for all these kinds of activities. So the active molecules in the immunoglobulin fraction are called as antibodies. So antibodies are basically nothing but the immunoglobulins against some antigens. So therefore their name is derived as antibodies. Because the immunity was mediated by antibodies which are contained in body fluids which were known at that time as humors, so this kind of immunity was called as humoral immunity. In 1883, even before the discovery that serum component couldn't transfer immunity, Elie Meshnikov demonstrated that the cells also contribute to the immune state of an animal. He observed that certain white blood cells, which he termed as phagocytes, they were capable of ingesting or phagocytosing the whole microorganism and other foreign material. Meshnikov hypothesized that the cells rather than the serum components were the major effector of the immunity, so therefore he termed as cell mediated immunity. It was shown later on that actually immunity requires both cellular and humoral responses. In 1950s, the lymphocytes were identified as the cell responsible for both cellular and humoral immunity. So experiments with chickens pioneered by Bruce Glick at Mississippi State University indicated that there were two types of lymphocytes, T lymphocytes and B lymphocytes. Now T lymphocytes have got their name because they are derived from thymus. So T lymphocyte mediated cellular immunity and B lymphocytes have got their name from bursa fabricius. Bursa fabricius is an outgrowth of the cloaca in the birds. So B lymphocyte mediated immunity will give rise to humoral immunity. That is, B lymphocytes will produce antibodies against the antigens. So this was the end of this topic, that is immunity. Thank you very much for your patience, for listening. Please like, share and subscribe my channel, and I am waiting for your valuable comments also. Thank you very much.